I'm Ayaantuu, and I'll be presenting with Gary and Keelan. The title of our presentation is Aren't You Glad I'm More Appealing with My Anti-Inflammatory Genes? And it's about anti-inflammation. So you can go ahead and start.
All right, to start with background and significance, the citrus sinensis, sweet oranges, they are known to contain anti-inflammatory properties due to flavonoids, and the natural cardioprotective and cancer-preventing bionutrient is what they are. They target inflammation in the immune system, and it responds to harmful substances that enter the body. And while this response is key in protecting the body, uncontrolled inflammation can lead to chronic inflammatory diseases such as asthma, peptic ulcers, Crohn's disease. The leading cause of death in the world is actually due to these chronic inflammatory diseases. And in a 2014 study, almost 60% of Americans did suffer from a chronic condition like this. So what we're proposing is to enhance the flavonoid content in oranges, a popular food item. It would not only provide people a safe and natural alternative to anti-inflammatory drugs. Long-term use in large doses of ibuprofen may cause stomach ulcers and cause GI tract bleeding due to these anti-inflammatory drugs that are currently available on the market. And then in figure 1, you can just see the molecular structure of a flavin 3 OLS as found in our orange. Biosynthesis and pathway for flavonoids. So in figure 2, you can see we have outlined CHS, which is the enzyme involved in the synthesis of flavonoids that we are targeting to change in the genome, so we have higher rates of production of the flavonoids. And then according to the flow of flavonoid biosynthesis, the CHS enzyme plays a vital role in catalysis and regulation of pathways of biosynthesis. Previous research has shown that the plants that are formed further down. So to increase the flavonoid production, citrus sinensis will be genetically modified to increase the expression of the CHS1 gene, and that's one of the 2 known CHS genes for citrus sinensis. And we're going to enhance that with the promoter associated with high rates of expression. So then we're just putting in a with the copy of the gene and the CAMV35S promoter. That's our citrus sinensis via an agrobacterium. It is feasible and innovative because CHS1, the gene's already encoded in citrus sinensis in the genome, so the modification of flavanols has not been attempted yet, but similar experiments involving citrus fruits have been performed previously and successfully. In an experiment with Citrus jambani, aka the rough lemon, it was modified using Agrobacterium tumefaciens strains. It harbored the PJRM17 plasmid with the HAL2 gene to transform the lemon into having an increased salt tolerance in order for it to grow in harsher conditions. And because our gene is from the same organism, there's a greater chance of success for the integration of the flavanol synthesis pathways, making it more feasible. And it's also innovative because we are using the organism's own properties to our advantage. We're not putting in an alien gene that may or may not take.
Okay. So looking into the next step, the first step is that RNA will be used due to its lack of introns, and it'll be isolated from the citrus sinensis, which is our orange. And then after, to isolate the CHS1 gene, PCR will be used to produce a single-stranded DNA from the RNA that can be used for the PCR. And the PCR is used with primers that contain linkers to the restriction enzymes, and you can see them in the figure 3 to the right. Next, the CHS1 gene is inserted into the pgreen 20049 binary vector, which will be seen in figure 4, between the left and right tDNA borders by cutting the tDNA region at the restriction sites BSI, WI, and BSR-GI, which is also seen in figure 3. The DNA ligase will then be used to bond the phosphodiester bonds together to create a new continuous DNA strand. After that, the pgreen 20049 is inserted into the Agrobacterium tumefaciens using electroporation. The Agrobacterium tumefaciens cells are used, which include the helper PAL4404 Ti plasmid. After that, the agrobacterium tumeflations are plated on agar plates, and white and blue selection is used to determine if the vectors were taken up by the cells. And then a scalpel will be used to pick up the agrobacterium tumefaciens colony that successfully took up the pea green 20049, and a leaf from Citrus sinensis is cut with a scalpel. Continuing on, the seventh step is that the agrobacterium tumefaciens attaches to a plant cell wall and forms a connection between the two via a T pilus created by the Vir proteins. Then the gene is inserted into the tDNA region of the P green 20049 and is transferred into the plant cell. It's aided by the Vir proteins from the PAL 4404 helper plasmid, which chaperones it. Once it's inside, it is randomly inserted into the plant's genome. Then the treated leaf is cultured on an agar plate so it forms calluses, and the calluses are then removed and further cultured on agar plates containing the hormones needed for plant growth and kanamycin to select for transformed cells. Finally, once the calluses have rooted, they are transferred to pots and cared for until they grow into full orange trees. So this is our vector diagram, and in figure 4 to the left, you see the PAL 4404 helper plasmid. That only contains the Vir region, which are the genes needed to transfer the tDNA to the plant host organism in the agrobacterium. And then on the right, we see the binary vector, which is the P green 20049, and that contains the tDNA region, which is the gene of interest, and in our case, it's CHS1, and that will be inserted in between the two restriction sites, BSI-WI and BSR-GI. So for the extraction, we're gonna have the extraction of hesperidin flavonoids from the orange, which is the Citrus sinensis. So we're gonna use a high performance liquid chromatography and a UV spectrophotometer. We're gonna have a high flow rate of water set to 1 milliliter per minute. We're gonna have an injection volume of 10 microliters, and we're gonna vector the orange juice at 300 rates per minute for 10 minutes. We're gonna adjust the pH at 8.5 by 10% of sodium hydroxide, and we're gonna filter the juice through a 0.5 micrometer filter and analyze by HPLC.
And then to see if this was significant, we'll use a t-test to compare the means of the hesperidin volume between the plants that received the tDNA test group and then the original plants that did not receive any tDNA tests. We'll have a null and a test hypothesis, and the null hypothesis if there's no significant difference between the means of the test group and the control group. And then if the p-value of that is less than or equal to 0.05, the null hypothesis can be rejected. And then in figure 5, you can just see how we would conduct that t-test with our different standard errors and critical values, and then also our null hypothesis will be shown in figure 5.
I'm going to talk about the evolutionary context of the CHS number 1 gene. And looking at this phylogenetic tree, it tells us the evolutionary history of the CHS1 gene between synchronous organisms, which means the CHS1 gene is an ortholog, which means it has evolved from a common ancestor. And looking at this tree, it can be seen that the gene is common among many synchronous fruits, which means if we were unable to do the project with the synchronous synthesis, we can also do the same experiments with other similar organisms other than synchronous synthesis. And a possible consequence of the introduction of a modified synchronous synthesis is the chance of polyploidy and breeding with other orange species. It would cause an imbalance of chromosomes in synchronous species, which could also result in hybrid sterility and breaking down offspring. And for ethics, there's a potential for long-term health effects through genetically modified food consumption, including an increase in antibiotic resistance, allergenicity, and exposure to toxic proteins. Excessive flavonoid intake was found to act as a mutagen in high doses. The adverse effects of flavonoids outweigh their beneficial ones, which means it would be really harmful for humans. And this technology could be exploited by large corporations or governments. For example, like in the case of the Coca-Cola controversy in 2014, and another one was the Italian government exploited African migrant workers through profit-oriented orange plantations. And obviously, we are working with citrus synesis, which is a type of orange, and the exploitation would be happen or would be permanent to the project.
Also, for the ethics, for the first bullet point, caution should be taken when ingesting the flavonoids, at higher levels than what's obtained from a regular vegetarian diet, which is 68 milligrams, and research and clinical testing of flavonoids at a dosage of 68 milligrams or higher would be needed before the oranges can be used for consumption. And for the second bullet point, regulations would need to be implemented and monitored before allowing this technology to be dispersed to the general public. So for example, you could limit the annual amount of oranges produced and monitor the conditions that workers have in these corporations.
And what's more, neomycin, kanamycin, and G418, as mentioned in the method slide, are antibiotics within the tDNA of the plasmid being utilized to genetically modify citrus synesis. If it breeds with local plants, the antibiotic resistance may be conferred to the wild population, and it would be really bad for the wild population and the environment. And the proposed genetically modified citrus synesis would need to be approved by the IRB, which is Institutional Review Board, for performing on human subjects. And then maybe done in order to receive licensing as an OTC drug. Reviews should be conducted often by regulating institutions or individuals to assess exposure risks to the environment and risks due to human or animal consumption.
And then for future directions of this project, the genetically modified citrus synesis or flavonoid supplement that we concentrated from the actual orange could replace current supplements that are on the market, and it could also replace non-steroidal anti-inflammatory drugs. Other citrus fruits or different types of oranges could also be modified in the same fashion by adding CAMV35S promoters in front of their own flavonoid gene sequence to increase the rate at which the flavonoids are expressed. And if citrus synesis with an increased anti-inflammatory profile is successfully obtained, enhancements such as increased vitamin and fatty acid contents could follow suit. So if this project were a success, if we did increase its anti-inflammatory profile, we could likely edit other sequences such as fatty acids or vitamins. And so in this orange, there's CH1, CHS2, and CHS1 genes in the orange, and they all are involved in the protein synthesis that's involved in the biosynthesis of flavonoids. So you can see the different genes that we could have chosen, but this proposition is only using CHS1 to increase the antioxidants to further the expression of flavonoids with anti-inflammatory properties. And if this doesn't work, we could add the CH1 and the CHS2 genes to hopefully increase that expression a little bit more with similar genetic modifications. And here's our references, and thank you for watching. Okay.